Professor Tony Zador. Tony is the Alla Davis Harris Professor of Neuroscience at Cold Spring Harbor Laboratory, unless that's changed recently. He's also a co-founder of the prominent Cosine Conference. In the context of today's workshop, Tony's extremely interesting contribution is an altogether different approach to connectomics using molecular biology to map the brain. Tony, please feel free to begin sharing your screen to present.
Okay, let's see how this works.
Biology to map the brain. Tony, please feel free to begin sharing your screen to present.
Okay, let's see how this works. Am I sharing? Is it working? Can I get some feedback?
Looks good.
Oh, good. All right. Well, all right.
Yes,
so
it seems to work.
Good. So thanks for the introduction. Thanks for that first talk. Was fascinating. So I'm going to be talking indeed about a different approach to uploading the mind eventually to connectomics based on molecular approaches, and it's summarized here in the first slide. Basically, our idea is that we can use molecular biology to uniquely label all the cells in the brain, then grind up the brain in one way or another, and use high-throughput DNA sequencing to read out the projections and connections of the brain. I should say as a caveat right now that we haven't achieved that goal yet. It has the potential plus side of much higher throughput, but there's a lot of technology development that needs to happen before that works. We're getting very close to making it work in the mouse, and there are some pretty more fundamental, but perhaps not insurmountable challenges to getting it to work in humans. All right. So I added one slide sort of in the context of this talk, and just wanted to give some thoughts about the problem of uploading a brain. I think the bad news is that it's going to be very, very hard to copy every neuron, every synapse, potentially every protein. And even if we did that, even if we were able to measure all of this, it would be very challenging to know what to do with that. Would we make a huge simulation? If we made a huge simulation, we would probably have to have very strong opinions about which of those components was important in order to sort of make up for the properties we didn't measure. I think the good news is that maybe we don't need to do it exactly that way. So I'll point out that the brain that each of us walks around with today is not the same as the one we were born with. It's not the one we had 25 or even 5 or even 1 year ago. And yet we have this illusion of identity continuity. We feel like we're at some level a continuous extension of the person we were when we were born and 5 years ago and certainly yesterday. And so I think that suggests that there's a path to uploading a brain in which we introduce some kind of a system that interacts with and mirrors our brains during life. And such a system would have the potential to sort of represent the substrate for a permanent uploading of our minds into potentially a very different kind of a system. But I'll say that I think we have little hope of uploading a brain if we don't understand many of the fundamental principles of brain function that we absolutely do not yet understand. I would say that we know a great deal about individual neurons and synapses, and we have very little understanding of how they work together to produce thoughts and feelings and identity and memories and consciousness. All right. So what we need to do is we need to understand simple circuits, and I like to sort of put a historical perspective on this and just remind people that the first person, I think, to really study circuits in the way that many of us are trying to do these days and tease apart the circuit was Descartes, who proposed a circuit by which a person withdraws his foot from a fire, a reflex, if you like. And the mechanism that he proposed was that the flames of the fire displace the skin, which pulls on a tiny thread. The thread opens a pore in the ventricle, and then the animal spirits flow from the ventricle, which is labeled F on the diagram there, through a hollow tube, and the animal spirits inflate the muscle of the leg, causing withdrawal. Now, many of you may recognize that our modern interpretation of what goes on differs in some ways. We no longer believe in the precise location of the source of the animal spirits F. But nevertheless, I think Descartes did make an important move forward here by actually writing down a potential circuit that we could, in principle, copy and use to build a brain. And I think that's the goal of many of us working on this. So why is circuitry important? Again, I don't need to belabor that point for this group. I'll just say that we want to be able to reverse engineer the brain. And to do that, I think we need to not only make a lot of measurements, because I do think those measurements are important, and many of the and develop new technologies to make those measurements, but we have to really think hard about how the circuits that we get out by making those measurements give rise to the kinds of things that would ultimately be needed to build a brain. And then the other really immediate need for understanding circuits is that most neuropsychiatric disorders arise from disruptions of neural circuitry. Autism, schizophrenia, depression, addiction, bipolar disorder, they're all probably, or many of them are probably in part, in large part indeed, caused by disruptions of either structural or functional circuitry. And the incredibly limited set of tools that until very recently we've been forced to work with have really limited progress, to the point that, as many of you may know, major pharmaceutical companies have largely given up the search in their own labs for discovering new drugs. And it's because what big pharma is good at doing is making a drug, not finding targets, and we don't even understand what those targets are. And that's really emphasized by the fact that really in the last decade or two, really the last decade, the major frontier in treatments of neuropsychiatric disorders have been in deep brain stimulation, circuit-based treatments that exploit the little that we do know about the organization of circuits. And so if we could know more about those circuits in mammals, in either humans or in models of brain function, that would be incredibly useful. All right, so why is circuitry hard? And that's shown here. The problem is simply that the axons are all tangled together. At least that's not simply, but that's really one of the big challenges. You see a bunch of neurons here, a bunch of wires. I'm not sure if you can see my cursor, but all those wires are somehow related to neurons depicted or shown here, and also neurons not on the screen. And we really need to be able to track them. Of course, with serial electron microscopy, that's increasingly possible, and yet that remains very slow. And the challenges are not just in the data collection, but in the data analysis. And although that technology is improving rapidly, I'm going to propose that there's a shortcut that complements the EM approach. Okay, so our core idea is to recast connectomics as a problem of high-throughput DNA sequencing by labeling each neuron with a unique sequence, a DNA barcode. And the idea is that way we don't have to trace neurons, merely read the identity of a neuron at its soma and at any place we wish to look, and we don't need to look at all the intervening sections. And that greatly simplifies the problem, and it allows us to exploit the tremendous improvements in DNA sequencing technology and more generally molecular biology. So why sequencing? Because sequencing is fast and cheap, and many of you may be familiar with this, with the fact that maybe aware of the fact that sequencing has gotten cheap, and yet it's still striking when you look at this graph to see just how quickly DNA sequencing technology has improved. So this is on the x-axis is time since 2001, which is when the human genome was sequenced, and on the y-axis is cost. And for this is cost on a log scale. So Moore's law on this is a straight line. Moore's law is a pretty healthy clip of improvement, and yet you see that the cost per genome has blown away Moore's law since about 2008. And so basically, any approach that you can sort of hitch on to the incredible improvements of DNA sequencing seems to me to be a win. All right, so right now what I'm going to focus on is the form of this technology that actually works really well right now in my lab today. We've set up a core facility. We offer this as a service now to collaborators, and that's projectome mapping. That's the mapping of long-range projections at high throughput at single neuron resolution. And to put this into perspective and motivate it, I'm showing here a slide from the Allen Brain Atlas. So what the Allen Institute did was a huge service to the entire mouse neuroscience community, which is they systematically went through and made injections throughout the mouse brain of a virus encoding GFP. So if they make the injection there on the right, they wait about 3 weeks or 4 weeks for the neurons that take up the virus to express it all through their axonal terminals. And then you can look throughout the brain, and anywhere you see green, you know that the neurons that you've injected send a projection. And they did this systematically, one injection per mouse, localized as far as they could to a single brain area. And then they, for each of those 1,000+ mice that they injected, they sliced the brain, took pictures, and put the pictures up online. And it's been an incredibly useful resource to all of us who work on this system. But that said, it has limitations. The challenge is that bulk injections don't distinguish single neuron projection patterns, and that's illustrated here. So on the left, you see an injection site. Those 3 circles are 3 neurons, and it could be that if you see green in the 3 target areas shown here, it would reflect the fact that neuron 1 projects exclusively to area 1, neuron 2 to area 2, and neuron 3 to area 3. Could also be, instead of a one-to-one mapping, an all-to-all projection mapping, or the brain being the brain. Most likely it's complicated, and that complexity actually is important to understanding what's going on. And that's the complexity that is missing when we use these kinds of bulk injections. And so the alternative to these bulk injections is to do single neuron fills, but that's incredibly low throughput. So here's a wonderful paper studying the projections from the cortex to the striatum in rats. For the purposes of this, what's important is that it's 1 neuron per rat. And this very nice paper showed the reconstructions of 25 neurons from 28 rats. This is a group that, at least at the time, it's a very skilled group. Since then, throughput has increased a little bit, but it's still not going to allow us to map all the neurons in a single brain at any throughput. So the inspiration for our approach actually came from a technique developed by Jeff Lichtman, Josh Sains, and published in 2007 called Brainbow. And there the idea is that to make the tracing easier, it would be convenient if the axons had different colors, if you could color code them. And there's this brilliant idea of coming up with a very clever genetic trick for endowing each neuron with a randomly selected collection of 3 colors drawn from a palette of about a dozen. And if you work out the combinatorics, given by the genetics they did, that led to a theoretical diversity of about 200 colors in principle. And that would actually be pretty nice. But unfortunately, color space is limited and hard to read. So although the theoretical diversity was 200, the number of resolvable colors that you could actually resolve with real microscopes, actually it's not even an issue of the microscopes, it's the overlap of fluorophores in general really is closer to, it's less than 10 generously, it's probably closer to 5. And that's shown here, where those three purple neurons are probably different purples, but they can't be resolved. So basically the core idea here is to replace colors with DNA sequences. And the reason is simply that there are a lot of possible DNA sequences. So we can kind of think of MapSeq, which is the name of the technique we developed, as a kind of digital rainbow where you replace colors with numbers and have each neuron express a unique sequence of DNA, a genetic barcode with a theoretical diversity that is essentially unbounded. And just to drive that home, there are 4 nucleotides, A, T, G, C. If you have a barcode, a random barcode of length 1, you have a potential diversity of 4. Of length 2, it's 16. And by the time you get up to 30, you get a potential diversity of 4 to the n. I'm sorry, of 10 to the 18, which is vastly more than the number of neurons in a mouse brain, a human brain, or for that matter, even in the human body. So a random 30mer is sufficient to uniquely label every cell in your body with very, very low chance of double labeling. More concretely, 10 to the 9 neurons are sufficient to label all 10 to the 7 10 to the 9 barcodes are sufficient to label all 10 to the 7 neurons in a mouse cortex with pretty high, with sufficient diversity so that the chances that 2 neurons will be labeled the same by chance are pretty small. And in practice, how do we do that? What we do is we've generated a virus. Right now we're working with a particular virus called Synbis. And viruses are generated in the lab from a plasmid. And so to generate a viral library, all we do is we take the plasmid that generates the virus, and we introduce these random sequences directly into the plasma, into the plasmid backbone. So we get a plasmid library, and then those plasmids give rise to a viral library of sufficient diversity. There's this remaining challenge of getting the barcodes to the synapse, because typically most RNAs don't travel to distant axons. And here in one slide, I'm summarizing the PhD thesis of a very talented graduate student in my lab, Ian Pycon, who figured out a modification of a synaptic protein that is shown on the far right that allows that protein, which normally traffics to axon terminals, to grab onto the RNA barcode through a so-called N-lambda domain. And it grabs onto the box B component of the RNA barcode that we put in and drags it out to the synapse. So our first proof of principle of this was to the locus coeruleus. This was published, I guess, a year and a half ago now. And what we did was we studied the projections of the locus coeruleus throughout the brain. It was known already that the locus coeruleus projects throughout the cortex relatively uniformly, but it wasn't known whether it was suspected and believed, but not really known, that the neurons in the locus coeruleus just kind of bathe the cortex in noradrenaline. So the locus coeruleus is the sole source of noradrenaline to the cortex. Noradrenaline is a kind of wake-up signal. It tells the brain to go on alert. And the usual view is that there's a cortex-wide alert signal that goes off when the locus coeruleus is activated. But alternatively, one can imagine that the locus coeruleus actually can specifically activate subparts of the cortex. So for example, if you need to specifically listen for sound, you might imagine that the auditory cortex is selectively turned on. And maybe if you need to selectively beyond the alert for light, it might be the visual cortex. So I'm going to skip over some of these details and just say that for the most part in these experiments, it turns out that the RNA acts like GFP. It acts like a bulk signal that bathes the brain. And the way the experiment goes is we inject in the locus coeruleus, that cross in the back there, and then cut the brain into slices. And then we extract the RNA from each of the slices and figure out which barcodes appear in each of those slices. And then we can demultiplex that and we can get a picture that looks like this, for example. So here is the profile, the spatial profile of projections for barcode 28, which corresponds to neuron 28. And the projection strength is measured in barcode molecules, which we quantify carefully through sequencing. And the x-axis shown here is the rostrocaudal position from those slices. So it's important to recognize that with this technique, the spatial resolution is determined by at the time of dissection. So we collapse all the barcodes that are found in whatever chunk of brain that we collect and put them and can't resolve them further. So this particular neuron you see has a very specific spatial pattern of projections. Another neuron, and that's at odds with the traditional model, another neuron has a much broader one, barcode 79, and there are even more complicated ones like bimodal projections. And in all, we were able to get 1,000 single neurons and their projections from just a handful of animals in just a couple weeks. So this is orders of magnitude more throughput for this system than has ever been. Well, since there were I think there were no locus coeruleus single neuron reconstructions, so more than orders of magnitude is infinitely more. But in general, 1,000 neurons in a handful of experiments is high throughput. We've since then, just a technical note, many of you are probably worrying about double infections. What if you get two neurons, two barcodes into the same neuron? And in the interest of time, I'm not going to go over why that's not bad, but it isn't bad, and you can ask me during the questions. What would be bad would be degenerate barcodes, insufficient barcode diversity that would end up with the same barcode ending up in 3 different neurons, and that would be disastrous. So we go out of our way to avoid that, and then we confirm that we avoided it. So if people have these technical questions, I invite you to ask me during the question period. I'll also say that more recently, we now have in collaboration with Tom Merzick-Flogel's lab, we did who did a bunch of single neuron reconstructions in visual cortex. We compared their reconstructions, 35 neurons in a couple of years, to some map-seq studies of those same projections, 533 neurons in a handful of experiments. And for the purposes of this talk, what's important is that they agree to essentially the resolution that we can determine in this experiment. And that there's a whole scientific story that I don't have time to go into, but that should be coming out in Nature sometime in the next few weeks, and it's on bioRxives right now. All right, so we have a core facility. This is up and running. We're now studying the projections in different animal models, different brain areas. And we're scaling up to do the whole brain in a single experiment, and I'm just going to go through that very quickly. We tile the entire brain with, or the entire cortex with barcode. Then we take much finer slices than in the previous work, align it to the Allen Brain Reference Atlas, and then we can project these back to Allen reference coordinates. And in a single experiment, in about a week for about $10,000, we can get the projection patterns of about 100,000 individual neurons. And for each neuron, we can see the areas to which it projects. So here are the strongest projections of 2 of these 100,000 neurons, just randomly selected neurons, to give you a feel for what kind of data that we have. So this is so much data, and of a different form, that although we've had these data now for almost a year and a half, we're still figuring out how to make sense of them and what to say about them. But we're writing this up right now. Nevertheless, none of this gets us to the connectome, so far I've only told you about the projectome. And so I want to, in the last couple minutes, talk about that. So the limitations of MapSeq are that first, it has crude spatial resolution, as I mentioned. It's limited by dissection. There's no notion of morphology or cell type. And I should say that I'm not getting any feedback on the screen right now. So I think I'm 25 minutes in, but if the MC wants to communicate with me, I'm looking at my cell phone. You have my cell phone number, and you can let me know how I'm doing on time or anything else. And if you want to communicate with me, I'm looking at my cell phone. You have my cell phone
Tony?
number. Yeah.
Yeah, you're fine. You're fine, actually.
Okay. If you want a side channel, I'm staring at my phone, so you can text me or whatever you want. Okay. In fact, that's reassuring to hear that you hear me, because for all I know, I've spent the last 25 minutes talking to my screen. I got to say, this is a strange forum where I don't see the faces of the people in the audience. You know. Anyway, so the second issue is that there's no notion of morphology or cell type gene expression. And my own background is in computation and in physiology. So the fact that anatomy is traditionally very hard to correlate or relate to neural activity, to knowing what the neurons did when the neurons were still inside the animal's head and the animal was doing interesting things, is distressing. And of course, as I mentioned, I haven't told you how we can get from the projectome to the actual synaptic connections that were needed to figure out the connectome. And the solution to all of those is a technique that we call barseq, that's modified from in situ sequencing, which is a technique that was developed in George Church's lab, and which we learned from them through collaboration, and then modified and adapted to the particular goals here. Okay, so to tell you how that technology works, I have to actually take you through just a little bit of the nuts and bolts of conventional DNA sequencing. Many people who do sequencing only have a vague idea of how the sequencing actually works. This is, when I say conventional sequencing, I mean conventional short read, high throughput sequencing as done by Illumina. And so this is how it works. On the left, you have a tube full of DNA, little bits of DNA that you want sequenced. You wash it onto a so-called flow cell. Those little black bars sticking up are sequences that grab onto the purple DNA that you want sequenced. And then the next step, you amplify each one of those single pieces of DNA into a little cluster on the slide of identical bits of DNA. So that's done by bridge amplification. And then the key thing is that then the sequencing actually becomes an imaging problem. So you take a snapshot of those clusters, but before taking the snapshot, you label each cluster with the next nucleotide in its sequence. So again, I'm not sure if you can see my pointer, but for example, this one in the left, the first nucleotide in that sequence might be a C, and so it's labeled with a fluorophore that's white. This one on the top is an A, so it might be pink. Same with this one, and this one here on the right is a T. Then you take a picture of this using originally they used a spinning disk confocal. They've actually changed that technology, but originally it was with essentially a confocal microscope. Then cleave those fluorophores off and add the next nucleotide. And so to build up the sequence of DNA associated with each one of those single molecules really is a matter of taking a bunch of pictures and aligning those pictures properly. And so the number of dots per picture represents the number of molecules that you're sequencing, and the number of cycles tells you the length of the DNA that you're able to get. So in this case, C, T, T, those are the first three bases, and then some dots. And typically these technologies can nowadays go up to several hundred nucleotides. So the key point here is that conventional high throughput next generation sequencing involves taking a picture. So for in situ sequencing, we can do essentially the same thing, except that instead of first extracting the DNA, we first immobilize the RNA in the cell, and then using a different set of molecular biology tricks, we convert each one of those individual molecules of RNA into a ball of cDNA, complementary DNA, that corresponds to the sequence we're interested in, the RNA. And from there on in, it's essentially the same problem of imaging. So here's an example that I hope will clarify what I'm talking about. This is a picture of a bunch of neurons in culture. The bright circles are cell bodies. I'm showing you these pictures from cell culture because the pictures are prettier, but we also do this routinely now in slices. And so what you see is, here's the key for reading it out. Each neuron is full of one barcode derived, or in a couple cases, two barcodes, but typically one barcode derived from infection by our virus. And just to show you how this plays out, here are two neurons that I'll show you the sequence of. The first base is an A, then we cleave that off, the next base is a G, and so on. And so by taking a series of pictures, we can read out the sequence of barcode in each neuron, and with different optics and modifying and optimizing a couple of things, we can also do it in axons and dendrites. So we're now applying this to study the projections in auditory cortex, and in the interest of time, I'm going to skip over most of that and just say that here's a picture of about 1,500 neurons whose sequences we've determined in a brain slice. Well, we took these neurons, we took a brain, we injected the virus in auditory cortex, then we waited 2 days, then we did in situ sequencing, bar-seq, at the injection site, and looked at the long-range projections using conventional map-seq. And in that way, we were able to get the projections of cell body positions of about 1,500 neurons. And so for each one of those little dots, we can click on it, and I could tell you the strength of projections to the dozen or so areas that we've studied. And again, there's a whole story that we're working out here. We can combine this with in vivo activity. We first use 2-photon imaging to figure out the activity of a bunch of cells. And then ex vivo, we can find those same cells and then figure out their projections. We can combine this with gene expression. Let's skip over that. And then here's what I think which is what's most relevant to the focus of this meeting, which is how do we achieve synaptic resolution? And essentially, I've already told you the answer, which is that we combine the axonal and dendritic labeling with barcodes in this tangled mess that I showed you. And we can figure out what the two barcodes are, the two barcodes are of the potential processes that are the processes that are forming a potential synapse. We have to actually go one step further and figure out whether there is an actual synapse there. Currently, we're doing that by using a synaptic marker. Going forward, we have some molecular tricks for actually entangling the barcodes associated with the two processes so that a single ROLINE will form that includes the sequences of the two barcodes, but only when the two barcodes are touching at a synapse. So in summary, map-seq and bar-seq open up the possibility of combining connectivity, gene expression, in vivo function at high throughput in a low-cost way. And I'll just summarize by saying that if the brain were so simple we could understand it, we would be so simple we couldn't. And I can't end without first thanking my collaboratome, especially Eustace, Xiaoyin, Lee, and former lab members, including Ian Pycon, and also my fundosome, especially IARPA, NIH, and the Paul Allen Family Foundation for some very early funding for this. And with that, I will close. Paul Allen Family Foundation for some very early funding for this. And with that, I will close.
Thank you, Tony. Let's give you an applause. I have to say that was really, really fascinating, and that looks like it's something that is going to help us go leaps and bounds forward in terms of understanding the connectome, in terms at least of the projections of circuits, maybe even more depending on how refined this technique gets. I'm pretty sure that there are quite a few questions, and I'd like to start by asking the people here in the audience for a question, if there are any.
Can I just ask?
Oh, just 1 second.
I think I'm in this weird mode right now of infinite regression.
Maybe you could just stop sharing your screen. That might be the solution for that.
So I should go to just chat. No, if I just toggle. Oh, okay, there we go. Thank you.
Okay, good. Yeah, sorry, Daniel, you had a question. Oh wow, I'm not sure if I even understood the question correctly. Could you say that? Maybe explain a little bit more.
I can't hear it. Or
so, yeah, I know. I'm going to have to repeat the question for you. This is how it's going to work from the audience here. Yeah. Mm-hmm. Ah, okay. Right. Okay. So Daniel is asking, now that you're doing transfection, now that you're putting things in the cell, what about putting in proteins to do something like a kind of computation, to record something else, to identify other things that are going on? And this
you mean like
this sounds like
DNA ticker tape?
Like the DNA ticker tape, sure. Yeah, basically.
I mean,
okay, let me just bring you on. There we go.
I mean, we're you know, the simplest version of that in principle is to combine it with calcium imaging. Right now, in the current form, there's a technical limitation in that the virus we use to do the MAP-seq is actually fairly toxic to the cells, and so this is a terminal experiment. We're in the process of switching over to a different virus, AAV, that should be compatible with calcium imaging, chronic calcium imaging. We have basically right now the way we do it is we do calcium imaging. Then when we're ready to end the experiment in the mouse, we inject with the toxic Synbis MAP-seq virus, get the wait 2 days, and then sacrifice the animal. A somewhat more convenient way to do it would be to combine the AAV calcium imaging with the barcode. And that's certainly on the to-do list, and we're working on it. But in general, anything that you can, I mean, it really, the compatibility right now is limited only by our choice of virus. In general, anything that you can, I mean, it really, the compatibility right now is limited only by our choice of virus.
Hi, Tony. Yeah, thanks for that answer. Just to clarify, if you hear yourself afterwards, it's because the live stream is a few seconds late.
Sure.
And so then I'm opening up the microphone for you to hear that, just so that you know that, you know, we're still listening to your answer.
Got it.
Yes, we have another question from the audience here. 1 second.
Well, Church is working on some like molecular recorders using CRISPR. Do you know much about that technology and whether it can be integrated?
Okay.
So the idea is to look at temporal synapse firing.
Yeah. Okay, the question is, George Church is working on some molecular recorders using CRISPR, et cetera. So this is very closely related to the previous question. And what do you know about that and about the, what was it? How it would complement your method?
So as far as I know, George's lab is working on is or has worked on 3 different things. So the very first thing that they were working on, which Adam Marblestone, as far as I know, George's lab is working on. Can you turn off my mic because I'm getting 3 different things. So the very first thing they were working on, which Adam Marblestone, confusing to me. Anyway, so the first thing that they're working on, that Adam Marblestone worked on, was the DNA ticker tape. And I'll let Adam discuss what the current and future status of that is. The second is that George and I are part of an IARPA consortium to apply this MapSeq and BarSeq technology to achieve single synapse resolution in the same IARPA project that Ken was talking about earlier for figuring out the connectivity of the visual cortex. So on that, we're full collaborators. My lab is applying this technology in all sorts of other ways, and I think, as far as I know, George's is in the context of neurons only focused on the ARPA, although a lot of things go on in George's lab. And then there's the third component, which is that I believe that they, as well as several other labs, have come up with CRISPR approaches to barcoding neurons in barcoding cells in the germline. They published one version of that. There are several other groups that have published different versions of that. Doing it in the germline has advantages and disadvantages compared with doing it with viruses. The advantage, of course, is that you can be confident that every cell has one and only one barcode. The disadvantage is that you limit yourself to working with a transgenic line. And so, because we're particularly interested in applying this to other model systems, mice, all sorts of rodents, marmosets, and potentially eventually humans, although that's a different discussion how that would work. We're actually pretty excited about the viral approaches, which turn out to be also more convenient. We're actually pretty excited about the viral approaches, which turn out to be also more convenient.
Thank you. We still have time for more questions, so now I'm going to grab a question that came in through the question and answer form online. Rose, or Rose, is asking, can DNA barcoding keep track of all the multiple connections of a neuron's dendrites and their strengths?
So we hope that, so there are 2 questions, I guess, embedded in there. First, can it track all of the connections? And using this spatial readout that I was describing based on BarSeq, the answer to all is yes, subject to technical limitations. The second question is, can we read out synaptic strength? That remains an open question in the same way that it remains an open question through all non-physiological approaches. I'm a physiologist by training, so for me, synaptic strength is defined by a slice experiment in which you record the synaptic connections between a pair of neurons, and you stimulate one neuron and record in the other neuron. There's a lot of data from EM, actually some of it, I think some of the first work from my own lab, the lab that I was a postdoc in, Chuck Stevens' lab, showing that there's a pretty good correlation between synaptic strength determined in that way and EM, the size and other structural components of a synapse determined by EM. And there's been, I think, a lot of follow-up experiments showing that there's a good correlation there. I think going forward, we can exploit some of that and also exploit the fact that there are molecular determinants of synaptic strength that we can access using in situ sequencing. So, for example, right now in a separate project that I didn't describe here, we are actually using a related set of approaches to figure out which synapses have recently undergone plasticity, where we use as a marker of plasticity the recent insertion of GluR1, type 1 glutamate receptor. So we're doing this in collaboration with Robert Malinow, whose lab worked out the basic mechanisms by which synapses, by which LTP works, long-term potentiation. Turns out that the insertion of a GluR1 receptor is a very good marker for synaptic plasticity. So right now we're using essentially the same set of, or we're trying to use the same set of approaches to figure out, if you like, the LTPome, the set of, the subset of connections that have recently undergone plasticity in the context of an animal learning something. So I think there are a couple routes to figuring out synaptic strength, either sort of directly structural or combining structural with molecular. So I think there are a couple routes to figuring out synaptic strength, either sort of directly structural or combining structural with molecular.
Thank you. I have another question from the online forum. This is by Keith Wiley. And he asks, can DNA barcoding be combined with a sort of slice and imaging that Ken described in a single subject, a single brain, or must we choose one or the other toward the connectome reconstruction goal?
So the question is, can we combine this technology with some kind of serial EM type approach? We've talked to various groups about that. Certainly, it would be awesome if we could figure out a way to do it. I don't think it's impossible. So I'm not an expert in those kinds of approaches. So I don't think it's impossible. It would clearly be just spectacular if we could do that. But so far, we didn't find a straightforward enough approach that would sort of, where we could crank out a proof of principle with, half a postdoc's time in 6 months, which is sort of at this point the level that we'd need to. I think that from what I understand, there are some routes that we could pursue, but we'd have to really double down and commit to pushing it. And so far, we haven't found the resources or the collaborator to do that. So I hope that the long-term answer is yes, and the short-term answer is not yet. So I hope that the long-term answer is yes, and the short-term answer is not yet.
Thank you. I think we have time for maybe one more question. So let me see, first of all, if there is one in the audience here. Okay, Daniel has one more question.
I was going to ask, is there any implications of this Neuralink versus infecting humans starter?
Okay. Oh, this is a tough one. Okay, so Daniel asks, are there any implications to development of this technique that are relevant to, say, startups like Kernel and Neuralink that are hoping to bring things to humans fairly quickly, or is this just a non-starter for use in humans?
So that's an interesting question. So, obviously the first answer is that anything we learn about the fundamental principles of brain wiring and connecting that with function is relevant to all these groups. So one route for getting there is that this is, we think this is, and I think the NIH also agrees, that this is probably the most efficient way to combine, to sort of achieve the Rosetta Stone goals that Adam Marblestone described in a bioRxiv paper a couple of years ago. Basically, to combine in a single specimen at single neuron, single synapse resolution information about gene expression, long-range connectivity, and function. And a lot of those components are probably preserved. So if we want to understand, for example, the circuitry, the subset of neurons, for example, that project from one brain area to another that might be particularly important, let's say, in alleviating depression or even in driving motor learning, this is potentially the fastest route to get there, certainly in a mouse. And once we have a pretty good idea of what the answer is in a mouse, we might know, we might have a better idea of what it is in a human. So that's one answer. I think the other, so I used to say it's a non-starter to ever apply this in humans because of the ethical issues. And I would point out that this is a destructive technology and that trying to label up a bunch of human neurons with viruses might be challenging to get through an ethical review board. And at one of the talks that I presented this idea, a neuroethicist came up to me and said, well, actually, it's not inconceivable that you could get approval to do this in a terminally ill patient who's basically going to be unplugged in the next couple days, if, you know, under certain circumstances. And similarly, one could imagine doing it in brain slices ex vivo. So we're not actively pursuing that right now, but I think if this technology matures a little bit, it may turn out that it's not absolutely inconceivable that it could be adapted. And there are some really crazy ideas, just finally, that we've toyed with that might allow the barcodes to propagate even under certain ex vivo conditions other than slices for a whole brain. So I guess that those are the routes that I see right now. So I guess those are the routes.
Thank you again very much, Tony. This was
Thanks a lot.
a fascinating talk.